All right. Hi, and welcome to the next webinar in our Plantae webinar series. My name is Jason Padilla, and I'm your technical host for today's webinar. Next slide, please. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I would like to give a special thank you to all our ASPB members who are attending today's webinar. Your ASPB membership dues help support making this webinar possible, so thank you. For any of you who have not yet joined ASPB, you can join today and use the discount code %10 to receive 10% discount on registration. ASPB member gets access to a lot of cool ASPB benefits, so you can learn more about ASPB and all the opportunities we provide at aspb.org. Next slide, please. This webinar is being recorded, and if you're looking for more great plant science content, please check out the Plantae YouTube channel, and you can find many, many more other awesome webinars there. So be sure to check it out. Next slide, please. Before we get started, I'd like to go over some housekeeping items to make sure that you get the most of your attendance of today's webinar. If you are experiencing technical issues, please let me know about those using the Q&A box, or you can email me at jpd@aspb.org. If you have questions for our guest speakers, please add them also in a Q&A box. Our moderator will be sharing them with our speakers during the Q&A, and so we'll try to get those answered. So if you have questions, feel free to drop them in the Q&A box. If you have trouble connecting or need to leave the webinar early, as I mentioned, we are recording this webinar and will be made available in the Plantae YouTube channel. So be sure to follow the Plantae YouTube channel to receive notification when new content are posted. And so without further ado, I will now turn it over to our moderators, our Plantae Fellows, who will facilitate today's session. With that said, take it away, Plantae Fellows.
Hello. Good morning, good afternoon, and good evening, everyone. Welcome to today's webinar, Figuring It Out, Creating Figures That Communicate Science. In scientific research, figures play a central role in how results are interpreted and understood. And very often, figures are the first point of engagement for readers, reviewers, and audiences, and they strongly influence how these scientific findings are perceived. Clear and well-designed figures can effectively communicate complex results, support scientific arguments, and improve the accessibility. So today's webinar focuses on how we can design figures and present the data more reader-friendly that communicate science clearly, accurately, and effectively. Next slide, please. To briefly outline today's program, we will begin with the introduction of the moderators and speakers, followed by the short talks from each of our speakers. We invite the audience to post their questions in the chat throughout the talks. These questions will be addressed during the Q&A session after all speakers have finished. Next slide, please. So today's webinar is moderated by 2025 Plantae Fellows, Elisa from Sant'Anna School of Advanced Studies from Italy, Elise from Syracuse University in the United States, Irene from University of Delaware, and myself, Krishna, from University of Southern Queensland, Australia, joining from your other side of the world. We'll be facilitating the session, moderating the questions in the chat, and guiding the discussion during the Q&A. Next slide, please. Yeah, today we are pleased to have Dr. Helena, Dr. Alicia, and Eva joining us as speakers today. Together, they represent different career stages from scientist, faculty to postdoctoral researchers and to PhD student, allowing us to bring a range of perspectives on figure design that aligns well with the diversity of our audience. Each speaker will be briefly introduced before the respective talks. With that, now I'll hand over to Irene, who will begin the introduction of our first speaker.
Thank you, Krishna. Our first speaker is Elena. Elena is a molecular biologist with a passion for design. After completing a PhD at the European Molecular Biology Laboratory, she worked with multidimensional data sets, images, and genome scale data at the Max Planck Institute in Dresden. With a scholarship from the Institute for Advanced Studies, she started researching biomedical data visualization practices. After working as an adjunct professor for bioinformatics in Berlin and a data visualization scientist in the medical faculty Dresden for 5 years, she is now an associate professor for data visualization in Chur, Switzerland. She remains a member at the medical faculty Dresden. With that, I welcome Dr. Elena. Please take the floor.
I need to share. So yeah, there we go. Okay, so I have 10 brief minutes today, so I would like to make the most use of it. So thank you very much for the introduction of what figure making is. That already encompassed it. I want to step back one tiny little bit and remind you that in life science and I mean also in your science world, everyone that is active as a scientist is in search for form and function. That's how we can describe life. And in search of form, we are very well acquainted with microscopy. So if we look into our world, we use microscopes, we use photographs, or we just look and make drawings. But we document the form that we see, and we draw conclusions from the diversity and from changes therein. As we go through collecting more and more pictures, and we realize that we can make summaries about them and actually get insight in a different way about function. So as we in my postdoctoral work, I expanded my single imaging to genome-wide screening, and I used many images to draw conclusions. So at that stage, I was no longer in pursuit of a form description, but actually a functional one. I wanted to understand the systematics of how shapes are gained, etc. And that is a point when you switch from visible science to invisible science, because data points are no longer visible. And as we are currently facing more and more sciences that are effectively invisible and just data points initially, we have to address in biology kind of suddenly in the last 10, 20 years how to make figures for such kind of data. So if you had a gel or a picture, it was easy. Now you have data points and you have to actually relearn how to make figures with that. And that's kind of the vast and growing universe of different chart types that we see in figures that are sometimes novel, sometimes imaginative, sometimes boring, etc. But they all convey the point about the data points that are otherwise invisible. And I want to remind you that this is really not only important for figure making, but much earlier, because every one of us, when interrogating such invisible data, needs to make it visible during the exploration stage. So in order to decide whether our control works or whether we have batch effects or if we have subgroups in our data, we have to make this invisible data visible with data visualization. So now I would like to talk a little bit about what data visualization really is, because that's the basis for all figure making it effectively. So there are a couple of basic principles that underlie the process of generating data visualizations. And the first one is abstraction, then finding patterns, finding suitable representation. There are rules how to do it, and how we can also apply rules from graphic design, in fact, to make them memorable. So what is abstraction? Abstraction is the process of looking into the real world, looking at a form, for example, and turning this into data points, which at the level of the computer just becomes zeros and one. It's very boring from a data point of what we can actually see. So we cannot see anything in this. We need to interrogate our abstracted data in order to actually make sense of it. And one way of doing that is to employ the human eye. And the human eye is fantastic at pattern finding. We are born to see, and we are born to respond to certain cues. One of them is color. When we are prompted by a certain color, we usually see it, and it evokes a different response in our eye and draws attention. But it's also absences. So you see a little gap in this data here, and gaps draw huge attention when there's a pattern that is otherwise systematic. And lastly, motion is also very powerful to draw attention. So there are certain cues that are inbuilt into our visual system that can help us to make sense of abstract data and employ pattern finding. And if you want to bring this to a more information level, these are kind of the cues that can be systematized about. So we can use color, orientation, size, etc. And if you want to learn more about this, the basis is in the Gestalt principles and the Gestalt form and how we see what we see. And the reason that I bring this up here is that all of these Gestalt principles or the basic patterns are used in the charts that we are making in our figures. And these are then representations that employ some of these pattern defining shapes. So when we go from our data points, abstracted data points, to a, for example, scatter plot, we are making use of, in this case, the principle of color and the principle of symmetry, because it looks like a line and we would see outliers, as well as the principles of XY position. We, of course, have a vast repository these days of chart types that we can choose from, which means that for any data set, in this case, we have a data set with just 6 data points, we can generate numerous different visualizations, and they each serve a different purpose in the communication. They can inform us about the anomalies in our data, or they can inform public about our finding. And it's good to remind ourselves that we have the vast repository to draw from, because that gives us opportunities to explore. And then it's important to remember that how we make these decisions is not a free form. It's not art. Data visualization looks a little pretty sometimes, but it's effectively information science and computer science. So that means there are certain rules of which data can be displayed in what manner. So if we have a certain data type and a certain message, then we have to find a suitable chart type. And we can do this, for example, with cheat sheets that are very nice to have, because they give us a first clue of what might be a suitable chart type for the kind of data that we had observed. And if we don't apply these rules correctly, we can end up with having figures, or also figures for ourselves, exploratory figures that are very misleading and might lead us to the wrong conclusion that experiment worked when in fact it did not, or it might mislead audiences to see patterns and data that don't exist. So we need rules of how we can pick data types. And you might remember the Friends Don't Let Friends Make Bar Plots initiative, which very effectively led to the abandonment of these bar charts, the ones that you see in the first chart. So these are given just means and medians, and basically they were used very prominently in biology. And in around 2016, when this initiative draw attention to the fact that it actually can hide non-normal data distributions, it became, from 2017, Nature Springer journals adopted the policies that these could no longer be used for statistical data. So there are rules, and we have to know them in order to start out correctly. Once we know all the rules and we know them well enough, we can of course also break them occasionally. So there are rules you might, if you go into the data visualization field a little bit more, you might have heard that there are rules such as don't use 3D effects, for example, don't use 3D pie charts, etc. And the reasons for this is, from an information design perspective, 3D effects in 2 dimensions don't work because they always hide something. But sometimes they can be very effective for drawing attention. We come back to that briefly. The next stage of data visualization is that we really need to be effective in forming memories. Data visualizations don't speak for themselves. They have to draw the attention to audience. So figures are not suitable for everyone. We have to make something visible in the figures, and it is our communication effort to do this correctly. So the first step is always measuring empirical layers. Second step is always analyzing and visualizing figures. And the third step is really making sure that it's read. Our paper is read by someone, our poster is looked at by someone, and it means something to the community. How do we form memories and data visualizations? We can look at art and draw inspiration. In art, you use beauty, you use humor, and you use horror to evoke emotional responses in viewers. And the very same principles are also applied in scientific visualizations if you look carefully. We see beauty sometimes in structural biology. This is from Dave Goodsell, who is famous for making these comic style representations of structures. And there's no wrong basis here, like structures don't have colors anyway. So whichever color you choose, it's up to you. So you might as well choose pink to decorate some aspects in the cell. There's also humor that really evokes knowledge about our world, and I hope many of you know the online comics XKCD, which makes use of the humor aspect. And then there's horror, for example, when we see horrible news about climate change and how dramatically the global surface temperature is rising, and the red is exemplifying exactly that. Climate change is very, very real. It's very dramatic, and we see this in the red, which captures the attention to that aspect. So we use beauty, we use humor, and we use horror, and we can also use it in small doses in the figures that we are actually preparing for our presentations or for our papers. And then I want to also remind you that just like with everything that we do, also fashion is very prominent in data visualization. So there's a recent paper that described the recipe for life. So we know for many years that 500 genes are sufficient. 21 amino acids are all that we need for life, 4 nucleotides make up the DNA, etc. And recently, this group that I collaborated with, they found that 2 lipids can make a functional membrane. And in order to present that data, the PhD student chose for this data a color scheme that's called Viridis, which is very, very popular. If you look through current literature, you'll find it everywhere. It has not always been popular. It's been recent, and if you look back, you will see that this anything that's published right now with color scheme looks modern to us. If the PhD would have been done 15 years prior, the color scheme chosen would have looked something like this, which was the default color scheme of ggplot2, and was very prominently used then in order to also say, I'm cool, I'm using R now, I'm no longer using Excel. Speaking of Excel, a PhD in the 2000 would have looked, the same figure would have looked like this, which was a default color scheme from Excel, and back when it came out again, it looked okay. In the 80s or so, and any time before that, figures were black and white. You had to pay for colored figures, so no one did that. So we used a color black and white scheme. And if you looked at PhDs in the 20s or 40s of the 19th century, bar plots looked different. They were actually it had no gap and were moved together. And in the century before that, we had black backgrounds with white bars, usually coming from, I guess, blackboards or something like this. So there is also fashion, and sometimes there is no rational choice for certain decisions, but it can look modern and current. And as PhD students, you want to do that. You don't want to look like you did your PhD 20 years ago. You want to say it's now. I want to remind you that, as I did at the beginning, that really figure making and data visualization is pervasive in our current day research. We need it for sense making from experimental data, whichever kind it is. And we need it for different purposes, for data exploration, for data presentation, and for data communication. Exploration is you sit on a computer and try to make sense of the data you obtained. Presentation is you go out to a conference or you write up a paper and show it. And communication is when you want to convince grant writers or committees, etc., of what you did. And depending on which stage you're at, you need to take care of different aspects. So for exploration, you need to know the rules in order to not mislead yourself. For presentation, you need to know the rules in order to not mislead your audience. And for communication, you just need to make sure you simplify your message enough. And when you had asked when you asked me to join the seminar, I think I'm done in 1 minute. Is that okay? Is that time-wise? Okay. I wasn't too fast. If you think of how can I apply data visualization for my figures, then I guess the reason that I'm invited to today to here was that I recently published a paper in Nature Cell Biology, which comes with a little checklist where you can go through the checklist in order to go from a shitty figure to a better figure, or from a great figure to a really, really great figure, depending on where you start. And the process kind of is summed up in the paper in a really simple way. You start out with your numerical data, which is then put into an exploratory graph. So whatever your software makes as a default, you go with it. And you then have to kind of make a lot of decisions on what to change in these default charts, such as removing unnecessary elements of the charts, removing boxes, grid lines to focus on the actual data points. Then replacing patterns to reduce or selecting suitable color schemes, making sure that the data is directly labeled so that you kind of go through to figures that are maybe more memorable than just black, white, and white ones. And you kind of, in the paper, I just outlined my view on the different steps that you have to go through and what you can do. And we can always work on 4 different levels. We can work on the chart itself. So is the data displayed correctly? But we can also work on the design aspect. So which color do I pick for that kind of chart and for that kind of data type? And then we can also make it legible. So we may have to make sure that the annotations are suitable, legible, and sufficient for sense making in our audiences. And I guess with that, I've exhausted my 10-minute slot, and I want to say thank you for the invitation, and I look forward to the questions.
Thank you. Thank you for that talk, Elena. With that, we'll go to our next speaker, which is Alicia. Alicia is a molecular plant biologist specializing in uncovering the molecular mechanisms that integrate hypoxia signaling with sugar availability. She's currently a postdoctoral researcher at the Santana School of Advanced Studies in Pisa, where her work explores the fine-tuning of ERF7 transcriptional activity through post-translational modifications and the role of mRNAs in plant responses to low oxygen. In addition to her research, Alicia serves as assistant features editor for Plant Physiology. She's passionate about science communication and collaborative research. Outside the lab, she enjoys reading, hiking, and baking. With that, I welcome Alicia. Alicia, over to you.
Thank you. I will now share my screen. Okay. Okay, so like that, I think we are all set up. Thank you very much, and thank you everyone for joining our seminar today. So I will be talking about the figure design, but my goal today is to give you an overview and a good basis for creating figures. I am not a designer, and I did not learn how to make figures in a formal course. Everything I know, I learned while doing science, usually under deadlines and one problem at a time. So whenever I did not know how to do something, I searched for it. I Googled it or I looked it up on YouTube because there are many free tutorials that help you solve very specific problems. And this is how most of us actually learn how to make figures. And the important message here is that you don't need to know everything before you start. You just need to know enough to solve the next problem. Once you accept that, the focus shifts away from tools and toward how you think about figures. And for me, a figure is not a collection of panels. A figure is a single visual object. And a multi-panel figure can work just like a puzzle. Each panel is one piece, and these pieces can be arranged in different ways, but they still need to fit together. If one piece is slightly off, the whole puzzle feels wrong, even if every individual piece is technically correct. This is why a figure is judged as a whole. Panels should work not only separately, but also together. And once you start thinking this way, something very practical happens. So if you invest time in designing one panel properly, this panel becomes reusable. It becomes a template. So you can copy it, move it, and reuse its structure across figures. And this is the important point. We are not talking, of course, about copying the exactly same graph and data. We are just talking about copying the graphical design. So, for example, here, these are three different independent Western blocks, but you can see that the way of presenting them is identical in both figures. And the same goes to these graphs. So different experiments, but the same planning and visuals. So once you create a properly looking panel, you are no longer redesigning everything from scratch. You are reusing a decision you already made. So one well-designed panel saves time everywhere. It's worth to spend more time because then you can use it not only in one figure, but in the next one and across all your papers. So at this point, people usually ask about software. Which program should I use? Which program gives me the best quality, etc., etc. And this is where I would like to be very clear. Tools are interchangeable. Principles are not. So you can make clear figures in a paid program like Corel, but you can also make them in a free one like Inkscape. And if these programs scare you or you don't have enough time to go and explore them, you can also make figures in something we are all familiar with, which is PowerPoint. These tools, of course, differ in access and convenience, but clarity doesn't depend on the tool. What matters are the decisions you make. And once the tool stops being the main concern, we can start focusing on some practical decisions that will form our workflow. And in this talk, I would just like to highlight a few of them. So the first one is the canvas size. Before adding anything,
Go from vibe coding to building securely.
okay, sorry. So before adding anything, you need to set the canvas to the final size. And either you can look it up at the journal page because many journals have their own specifications. But as a default, you can also set up the size of, for example, the A4 page format. If you design on a bigger canvas and shrink everything later, of course, the text will become unreadable and spacing will collapse. So the single decision affects everything else that follows and sets you up for a good start. Then the second principle is alignment. When elements are aligned, the reader's brain does not have to constantly reevaluate where things start, where they belong. It can focus directly on the data itself. Practically, it means that we should treat alignment as a default, not as a correction. So if so I align panels, labels, axes, and you should do it early on before adjusting individual details. So once things share the same edges and spacing, the small changes you do later become much easier. And these alignment tools that I highlighted here are useful because they let you be consistent without precise measuring or guessing. So you do not need, I mean, of course, you don't need a perfect symmetry, but you do need an intentional structure. I think a good rule of thumb here is this. If two elements are meant to be compared, they should be aligned. And if they are aligned, the comparison becomes effortless for the reader. And the third principle that I think is very important is, of course, the visual flow. So figures are read, not just seen. So if the reading order is not obvious, the reader has to work to understand the figure. They jump around with their eyes, they get tired, and at the end, they miss the message. So whether you organize your panels in rows or in columns, I prefer rows, but at the end, it doesn't matter as much as making the order clear. So if the reader gets lost, the flow is wrong. And before polishing anything, what I like to do is either print my graphs or put them then on a single piece of paper, or just organize them in PowerPoint to understand how the story is flowing. And something that we sometimes forget about is that even if when everything is done, there is still one step that can undo everything, which is exporting. And I consider exporting also part of the figure because you can design the best, the most well-structured figure, but then completely ruin it with poor export settings. So low resolution, wrong format, or excessive compression can undo all the work you put in. And at this point, I would like to step back from all the technicalities and talk about something more fundamental. So designing figures forces scientific choices, and this is actually, in my opinion, one of its biggest strengths. So when you start to make a figure clear and readable, you very quickly realize that you simply cannot show everything. There is just not enough space. And maybe this is not really a design problem, but a scientific one. But still, making a figure means deciding what is central to the story and what provides support. So, of course, some results need to be shown together because they answer the same question. However, others are important, but they can work better as context or confirmation elsewhere. So for me, this process of choosing is part of scientific thinking. And by designing the figure, you are clarifying what the main message is and what can remain in the background. In that sense, clarity is not about removing data. It's just about organizing and prioritizing it so the argument becomes visible. And with this, this brings me to my final message, which I would like to leave you with. So we have to remember that figures are not decorations. They are scientific arguments. They are part of how we think, how we decide what matters, and how we communicate science. So if your figures are clear, consistent, and honest about what they show, they are already doing their job. Thank you.
Thank you, Alicia, for that presentation. Now we're going to move on to our final speaker, Ava. So Ava is a PhD candidate and Unidel Distinguished Graduate Scholar at the University of Delaware, researching controlled environment agriculture and automated irrigation systems to enhance data accuracy and reproducibility in plant science. Her work advances irrigation management and data collection technologies for hydroponic and chamber-based crop studies. She developed a passion for plant growth system design in controlled environments during her undergrad studies and completed 2 NASA internships, first at the Kennedy Space Center studying plant growth in the International Space Station, and later at Langley Research Center advancing food production research for Mars missions. She was also part of a finalist team in NASA's 2019 Big Idea Challenge to design a Martian greenhouse, integrating plant science and engineering for sustainable extraterrestrial cultivation. Go ahead, Ava.
All right. I think. All good. Yeah, hi everyone. I'm Eva. I'm super excited to be talking to you guys today. Let me move this really quick, sorry. Sorry. I am really glad the other speakers talked about some of the more specifics with clarity. And so I'm also going to be going over, just again, a little bit more into methods figures, touching on the creating key message, adjusting for audience and media, and then going over an example and sharing some tools and resources. So methods figures, they can be really important, especially when they're used like as part of your scientific narrative, when you're wanting to really increase clarity about like what you did, you want your audience to understand what you did, when you did something new or created something new, and when you want other people to be able to follow what you did and reproduce your methods. So I recently attended a science communication workshop, and one of the stats they shared with us was really surprising to me. So the proportion of US adults that have confidence in scientists to act in the best interest of the public has dropped 10% since 2019. And Another thing that they had in the article was that like most Americans see research scientists as intelligent, while few say they're good communicators. And so I believe these facts are really connected. And so it's very essential that like we as scientists use all of these tools, but like principles when we're creating figures so that we can really increase this clarity for broader audiences, so we can share what we do in a way that more people can understand. Like has been said before, your key message, whatever it is you're trying to say, that determines what kind of figure you're going to make and what you're trying to emphasize. A lot of times for methods figures specifically, that's displaying novelty, again, improving clarity and like making it easier to reproduce. And generally just everything unrelated to that should be removed from your figures. Your audience is going to dictate your context and complexity. So you have to think about who's going to be seeing the visual. Is it your specialists? Is it people who work in your same field, your same lab? You don't need to have much context for that. General audience. So like I would consider all of you a general audience if I was to share my research. And of course, there's non-experts in the general public, which generally needs a more focused and simplified message, focused on like the most important part of your research. And so depending on your audience, you will need to adapt your key message and then determine like how much context do they need, how much realism, how much explanation, or how much detail, like for them to really understand what you're doing in your research. We kind of just need to demystify it. And so as part of shaping a figure to most effectively communicate your message, it's really, really important to shape your figure based on what medium you're using. And so if you're submitting a figure as part of a manuscript, the audience will be able to take their time, and you can use a lot more higher detail and complexity, and chances are they're going to have better background knowledge. In presentations, on the other hand, the audience only has a little bit of time to look at it. And so figures should be very simple. The emphasis should be clear. You can definitely use animations. I really like the morph transition if you're trying to break up complicated figures. And like higher contrast, it always sucks when you're sitting in a presentation and everything's washed out and you can't see what's on the screen. Let's see, with posters, you really want to have things be large, readable from a distance, text should be minimized. And so just general takeaway point, what works for a journal is not generally what works best for presentations and posters if your goal is to communicate effectively with your audience. And so they're just touching on these. I know the other speakers already went over them to some detail. And so just when you want to emphasize things, you can use visual contrast. Please do not use all of the methods of visual contrast in one figure. That creates a lot of visual noise and becomes like hard to understand. To increase clarity, as was already spoken about, like using clear information flow, generally top, bottom, left, right, not using things for decoration, just using things for the key message, explaining and labeling things when you have the first instance of an object, being consistent with colors and shapes and orientation, and then of course getting feedback from a lot of different sources, because that's a good way to tell if it's clear, if someone else can understand it. Next step is just getting the key visuals. And so depending on what you want to show, you may want to use photos, which are really important when you want to have evidence. And these can be paired with overlays to clarity and real world context. Photos also tend to have a lot of noise in them just because there's a lot more detail going on. And so sometimes you want to use illustrations, and that's when clarity is more important than realism. Illustrations also allow us to highlight a specific theme or element and show something that is invisible going on. And then just like a little bit more abstract, like using icons and using shapes, when whatever your process you're trying to show, it doesn't really matter what it looks like. These are also really helpful when you're trying to simplify for broader audiences or just reduce visual data and noise and to just focus on something important. And so if you're unsure kind of where to start, but you have some ideas in your head, often you can start with a sketch. And so it kind of brings it all together. If my key message was to show my plumbing configuration and water movement through a hydroponic system to an audience of people within my field, I might start with a sketch like this. And while this gives a really good idea of some of the physical parts of the plumbing system, and someone familiar with irrigation systems might be able to understand it okay, it doesn't really explain water flow. And most people would be relatively confused about what's going on or why it matters. And so if instead my key message, and it's like much nicer photo to show the dimensionality of the plumbing system, where the pipes would be physically in relation to each other, this kind of graphic is really useful. Because in real life, the pipes are hidden, unable to be seen. And this is also helpful for someone who wanted to recreate the system, or an engineer who wanted to understand what components are like used. And like using the color coding, you can see like where the water movement is happening. And like was mentioned before, some of this with 3D graphics, there's just a lot going on visually. And so if the configuration of the pipes didn't matter, I would probably not want to go with this. Because 3D elements like take a lot of time to look at and to like figure out where the pipes are. And so instead, I'd probably want something like this. And now if you look at this for any amount of time, you'll start to see some issues. And so, and this, again, just really goes into getting feedback. So for this, like this was saved as a PNG. Like she mentioned, I'm glad, like just on exporting, they can ruin everything. It's unclear where to start. It's not the top, it's not the left. It shows all the existing channels, but like all the labels are repetitive, and it's a poor spatial approximation with an inconsistent viewpoint. Like you're looking top down on the channels and you're looking at the side view for the primary reservoir. And so not awesome. And yeah, important to get feedback because I can promise you when I first made this diagram, I was not intending that to be the bad example. And so if you look at the updated one, this was saved as an SVG. It's clear things start in the left corner, there's reduced repetition, and it is less accurate to the system compared to the 3D model beforehand. But it is easier to understand where the water is going, all from like the same viewpoint. I also removed the vertical text because that can also make things harder to read. And so I just wanted to like highlight these tools that I used really quick. So for the 3D figure, I used Autodesk Fusion. It's free for students. Blender is another option. As they were saying about tools, this is really worth learning if you're planning on 3D printing. Otherwise, I wouldn't waste your time on it, just because, again, big time sink. And a lot of times, unless you specifically need to explain where things physically are in relation to each other, 2D drawing will work just as well. The other tool I used was Draw.io. So I learned from engineers, and it's very easy to learn, can import icons and images. It's better than PowerPoint for mind maps, like the arrows like stick to the objects and when you move them around. But yeah, just listed some other ones just in case you're unfamiliar. And so another tool I just wanted to highlight, so another student at the University of Delaware, Tanny Wiest, she created these with BioRender, and I thought they looked really good, and she was kind enough to share them with me so I could share them all with you. And so if you do this kind of work, I'd recommend checking it out at least. There's a free version and a paid version. I believe there is something about needing to have the paid version for publication, but your lab or your university might offer that. Of course, there are other alternates like Illustrator, Inkscape. And then I also included just some other like icon sources, just in case that was something that you were needing. But yeah, Google works too. Yeah, this was just like what the website looks like. And as you can see, they have a lot of free templates for a variety of plant science uses. Oh, and there's that again. The last thing I wanted to touch on really quickly was graphic abstracts. And so just like the same thing with the other methods figures, you like really want to have it down to your key message. The things that I found was to like summarize the biggest takeaway from your abstract, 1 to 2 sentences, and like get feedback on that before proceeding. And then your audience for this is going to be your generalists, like general plant science biology, aiming to be broad and understandable to support interdisciplinary work. And so Dr. Jamber actually wrote a paper also on writing graphical abstracts, and here were just some points that I pulled from that. So they're not intended to be a complete understanding of the work, and they are intended to attract attention and be read alongside your written abstract. But all the design principles, key visuals, tools for methods, figures, and honestly, what other people have talked about as well, they all apply and can be used here. And here are just some additional resources for the creation of graphical abstracts. And then to the last thing, I did a lot of reading up on different, like how to make visuals well in preparation for this. And so I just wanted to share these with you in case you were interested, because it took a bit of digging to find them. So yeah, that's all I have. Any questions? I'll stop sharing.
Yeah, thank you to Eva and all of the presenters for your really insightful and fantastic presentations. We're now going to begin the Q&A session. So if you haven't already, put your questions in the Q&A. In the interest of time, we'll try to get to as many as we can, but we'll try to pick some ones that are particularly relevant. The fellows and the speakers have already been in the Q&A answering some of the questions already. So especially those of you who are looking for paper links, things like that, they are in the answered questions in the Q&A, and those are there ready to go. But I think we'll start out with our first question. So we'll start with Helena. We did have a question really thinking, do you think scientists and students will need formal training in design in the future? And if so, how do you think this would be done?
Well, I like to differentiate between design and data visualization. Data visualization for me is an epistemic layer that is inherent in the scientific process, and it's just a method like making sure that your sample is correctly placed, or like designing a control experiment. Just like that, you need to design and engineer data visualizations that capture the structure of your data and that truthfully represent them to you and to others. And the design, I would argue, so I would not train biologists in a typical design stuff because that's often not rooted in the data visualization principles. So I'd rather they have better training in computational methods, which includes how to deal with statistics and statistical graphs, because color choices are not random. And even though something is good choice from the design perspective, we cannot simply use certain color schemes for certain data types. So I would argue that this should never be taught by designers, but rather by people who have training in biology and or computational biology and or bioinformatics and or computational sciences.
Great. And then I think Elisa is going to take our next question.
Yes, I have a question from the audience for Alicia. And the question is, in your experience, what advantages do Inkscape or CorelDRAW have over PowerPoint?
I think the simple answer is that is the quality, because Corel and Inkspace are the vector programs, so you can export your figures in a higher quality. So I would always say that it's more preferable to use them for big figures where, and by quality, I mean that if you export the data and then, for example, change the sizes, if you make the figure in PowerPoint, you can end up with text that is a bit blurry and not so sharp. So yes, in my opinion, the biggest difference is the quality and the details.
Okay, Ala, thank you very much. I think we have now a question for Eva as well.
Eva, you had mentioned kind of thinking about your audience and kind of different audiences and how you adjust for that. We had some questions about, did you ever work with engineers, and how are you able to communicate technical knowledge in biology across fields?
Yeah, so for I know I mentioned like the Martian greenhouse project, I was the only plant scientist on a team of engineers. And so I feel like communicating with them, a lot of it was, to some extent, I feel like they needed to adjust their communication to me a little bit more and just like simplifying things. And I had to get better at asking questions when it came to things that I didn't understand. And a lot of times, they don't have the same assumptions of the flexibility of life as with like working with biology. And so there were a lot of times like, oh, here's what's most efficient, or here's what's and I've seen like, again, so like engineering things, they have a lot less visuals, or they might be a lot more just like, here's the fundamental information. And it doesn't give the context like for broader audiences, or for like what plant scientists have, or like who might not understand or might need more information. And so, yeah, I don't know. It goes both ways. I don't know if that really answers the question, but yeah, as far as like visualizing things, when it comes to working with engineers, and that's where it's like I have learned some of these engineering tools like Draw.io and like Fusion 360. And so some of that, again, might just mean coming together, and depending again on how much you're working with them, like it might be worthwhile for you to learn those tools so that you can kind of communicate in the same way. So yeah.
Yeah, thank you, Eva. I think we're going to go back to Helena again. I think your comment in the chat about biorender and copyright may have surprised some people. So I think people were kind of just looking for kind of more information on that situation with biorender, and then what would be some possible alternatives to biorender.
The person who's really an expert on this is Simon Durr, who is a founder of BioIcons, which is a free alternative to BioRender. So he founded BioIcons because there are tons and tons and tons of icons that we have freely available under open licenses already on the internet. And he kind of just collected them. And he's a PhD student, by the way, as well. I was well, not anymore, but when he did it. And he brought that brought that point to my attention. So basically, currently, the license agreement with BioRender is that the copyright is maintained by BioRender, which means whatever you design technically stays under their own copyright, which is crazy because that does not apply to other software. So if you use Microsoft Word and the text that you write is still yours. It doesn't belong to Microsoft. Or if you use Illustrator, the drawing that you make with Illustrator, because you paid once and use it as an instrument, the figure is yours, the copyright one. And BioRender, that's not the case. So that's a really, really weird situation and something that was about to get solved until, so from NIH people who were really going after this, which is why they also created the NIH Art Collection to kind of give you guys in the US some free resources, which can then be used widely by other people as well. That kind of got stalled in the last 1.5 years, so no progress, unfortunately, but of course it's still an issue. So technically you cannot publish under open licenses anything that you created with BioRender. In addition, they want you to include the icon created with BioRender at the bottom of every figure, which is also really ridiculous because it looks ugly, but also you paid for it. So why would you include that icon in every single one of your words? You don't have a Microsoft icon in every single of your papers as well, right? That makes no sense. So they really have a problem with licensing. And I think we just pay too much money. It's like the license per person is what, $200 for a year, something like that. And if you have a several seat option for the lab, that's more than 1,000. It's just really expensive considering that all you get is free icons. Yeah, that's the end of my rant here.
Thank you so much.
Thank you, Helena. I think we now have another question for Alicia. And the question from the audience for you, Alicia, is you emphasize that figures should serve as arguments, not decorations. Nowadays, we often use different AI tools to make figures more attractive. What do you think about this practice?
So I guess we can continue ranting because I am not a fan of using AI, especially for graphics. I find it unethical, especially when you create something from zero, because usually it steals from artists. However, I also understand that we need some help maybe when the task is overwhelming. And we have to remember that AI should be used as a tool, not to replace our thinking and our planning. So if you feel insecure or you have no idea how to structure your figure, okay, you can discuss it. If you don't have another human to discuss with, you can discuss it with AI. But I would not use it to create the figures. At the end, AI is prone to create some weird things out of nowhere, and I think nothing can replace human touch. So if you want to create a schematic, just think about how would you explain it to your siblings, to your parents, or whoever who doesn't know the science behind, and I'm sure you can manage without AI. So yes, maybe I'm the wrong person to ask because I am very much against using AI. And I think Helena will have another comment on that.
If I may, yeah, I agree. Like for full schematics, it's like super error prone. Like something is always wrong, so it's not really worth the effort. What I've made very, very good experience with is actually designing an icon, because if you go to the big icon libraries, of course, you have something really specialized for your plant research or something. Then, of course, you can download the SVG file, the editable file from Noun Project, also for free, and kind of edit this and then publish it as your own. This is as bad for the designer effectively as just telling AI to design an icon for you as an SVG. So I've used it for creating icons that I just couldn't find, or where the style was really just not matching. And I guess that has proven in my hands to be quite efficient. And I really personally don't see a problem there, because if I would use the same icon downloaded on the internet, and I'm also repurposing or making a collage, as we call it in art, other people's work.
Thank you so much. And we got like a bit of discussion there going. Always interesting to see like kind of the different views on somewhat new and controversial subjects with that. I think kind of jumping off the tools idea a little bit, the next question for Eva, you had mentioned multiple tools throughout your presentation. We had some questions about like which tools would be best in certain situations, and then also which tools would be best for students making effective figures.
So honestly, so I did list some in my presentation. I'm guessing that'll be shared out, but if not, just as far as icons go, yeah, like was already talked about Noun Project, bio icons. I saw some others listed, SVG Grepo, and SVG Grepo, sorry, Reactome, EDI, Philoptic. For arranging, PowerPoint, Dry.O, Illustrator, Inkscape. Honestly, I think one of the best ones for students tends to just be PowerPoint, just because there's a lot of, it's like people are already generally familiar with it, and it's not like that much of a skill jump or time investment to like learn how to use the alignment tools, learn how to like have things sized to the proper like specifications of it, and just kind of like, and also like having a palette and having all of like that already set out. If you're going to be specifically doing a lot of like network maps kind of things, or like really like more intense like input output processes, or like code diagrams or anything, then that's where I think like Dry.O would be a really good one to introduce. And it might be one just to throw out there for people who might be interested. You can still import all of your images or icons or whatever you want into it. Yeah, 'cause it has the grid, it has really like the arrows, yeah, stick to objects when you move them around, which PowerPoint doesn't always do. And so, and it has like a lot of like the same alignment features as grouping features. As for like Illustrator or Inkscape, those definitely just take, again, a little bit more of a learning curve to get familiar with. Kind of like once you get familiar with, they're okay, and they can be like good like skills to use and good things to use. But yeah, just like I would expect, yeah, it's more of a teaching time would need to happen for that, as opposed to just using PowerPoint or even Dry.O. So be my general recommendation. Yeah, if you want to do 3D stuff, there is a lot of tutorials for Fusion 360. It's free for students. Blender, I haven't used it, but my understanding is that it's free and open source. And I know that there are programs that do and like have created 3D molecules and stuff. And so maybe that's something that you'd want to explore. And my understanding is that's pretty user friendly as well. So yeah.
Okay, so thank you, Eva, for answering the question. In the interest of time, we are stopping the Q&A session, but I still hope that it provided answers to many of your questions. So as we come to the end of today's webinar. I'd like to thank all of you for joining us today for this discussion on how figures can truly communicate science. And we are deeply grateful to our speakers, Dr. Helena Jambor, Dr. Alicja Barbara Kunkowska, and Eva Wirtel for joining us today and sharing not only practical strategies, but also a way of thinking more intentionally about visual communication. As today's session reminds us that figures are often the first, but sometimes the most long-lasting impression of our work. And when designed thoughtfully, they can clarify complex idea, guide interpretation, and extend the reach of our science, which is maybe the most important thing. So I'd also like to thank the other Plantae Fellows that helped me organizing the webinar, and Jason for his support with logistics, technology, and promotion. We hope that you leave today inspired and that you will join us for future Plantae webinars that you can find on Plantae web pages and social media. And we want to wish you a Merry Christmas and a wonderful rest of your day. And Jason, now the floor is yours for closing the webinar.
Thank you, everyone. Again, a big thank you to all of you, especially to our speakers, our moderators, and to all the ASP members that joined today. We hope that you find this webinar really valuable. And as Eliza said, we'll have many more. And so we hope that you tune in to the next 10. And so with that, I hope you all have a wonderful holiday season and a very, very prosperous new year. And we'll see you on the next one. Have a great weekend and see you all next time. Bye, everyone.